Stanford University.
Good afternoon, everybody. I'm Brian Wendell from the psychology department, and I'm also involved in electrical engineering and ophthalmology and radiology. I'm the third of Bill's deputies. You're only going to get to see three of us today. Malenka, who we never know where, yeah, he's Malenka somewhere. The one thing you can depend on with Malenka is he's rooting for the New England Patriots, whoever he is. That's a constant for him. And some of you may wonder what these deputies actually do. We all meet, it's a very pleasant group. We all meet, Miriam, sometimes Rob, often Scott. Once a week on Monday afternoons from 3 to 5, we eat some peanuts, and we help Bill figure out what he needs to do next. And Bill does the overwhelming amount of work with help from Tanya and so forth, and the rest of us cheer him on and give him occasionally okay advice. But I'm not here to talk about Bill. I'm here to talk about Nico. Nikolaus Kriegeskorte is here. It's my pleasure to introduce him for this afternoon's talk. And he joins us from Cambridge, England, where he is a program leader in the memory of the memory perception group at the Medical Research Council. And he works in cognition and brain sciences unit there. Nico has actually worked with a large number of people who are really kind of friends of Stanford, and I see Nico is coming from this wonderful tradition. He originally got his degree in psychology and computer science, which is a great interaction, something that we here in psychology are always very positive about. I see Jay McClellan there, and he's really leading an effort on that. Nico then took his degree with Rainer Goebel, who's here all the time, and a great friend of the university, and that was during the early days of the development of functional magnetic resonance imaging. And so Rainer got you pushed off in the direction of confronting brain data and actual human measurements at that Max Planck there. He then went on to work as a postdoctoral fellow with Camille Ugerbill, who's been longtime friends of ours here, and with, I see Gary over there, and we see Camille all the time. And then he went and took another postdoc with Peter Bandettini and Leslie Ungerleider at the NIMH before going on to Cambridge. Nico is one of an exciting group of young investigators who are using computational methods to investigate how information is represented and processed in the primate brain. And there will be a fair amount of young people talking this afternoon. I'll remind you about this later. And his lab's focus is on the neural mechanisms of visual object perception, and both health and disease, and how these mechanisms vary between individuals. So looking at one person at a time and understanding individual differences. And Nico, thank you for coming out and making this trip. We look forward to your presentation.
Thanks, Brian. Wow, what an honor to speak here. Let me see if this works. That's great. So I've adjusted my title a little bit to make it even more provocative than it was before, to make it outrageous enough to, I hope, provoke you to ask me some difficult questions at the end. I love the fact that there's enough time here for actually having some interaction at the end, and I hope that's going to work out and you're going to challenge me on some of the things that I'm going to say. So my title is Deep Neural Networks, a new framework for understanding how the brain works. What does it mean to understand how the brain works? I think this is a question that has been of great interest to me over the years, and it's only in the last couple of years that I've kind of come to an answer for myself. I don't think there's one correct answer to this, but I'll give you my answer, and maybe you have your own. So to me, it means to explain neuronal activity patterns, to explain behavioral data, ideally in humans, because ultimately that's what motivates me. I want to understand the human brain ultimately. Using fully explicit computational models, so models that are implemented in computers and that actually perform the task, the cognitive task in question. Ideally, these models should be neurobiologically plausible, and they should perform real-world complex cognitive tasks. So not toy versions of tasks, but they should be able to interact with the real world. So they should be artificial intelligence systems at the same time. So historically, different disciplines have tackled some of these criteria for success in my mind, but we haven't really been able to combine all of them at the same time. So for example, behaviorism was concerned just with behavioral data, and that was a major limitation. Cognitive psychology stretched in the direction of information processing models of what's going on inside the brain, but its verbal cognitive theories fell short of being fully explicit, and that's probably the reason why it was superseded by cognitive science, which was at this cognitive level, but more rigorous about the modeling. It had fully explicit computational models. And I think that's why cognitive science, in my mind, is still a relevant discipline. And the Cognitive Science Society conference that initially got me excited about brain and cognitive sciences is a good conference to this day. However, cognitive science doesn't really engage brain activity. So when, for me, it came time to choose a PhD project, I felt like AI and cognitive science are not enough. I want to study brain activity as well. So we had cognitive neuroscience at the time. So cognitive neuroscience, with the advent of brain imaging, non-invasive functional magnetic resonance imaging in humans, that was a revolution, and it was very important. However, as we tackled these complex brain activity data sets, we temporarily lost theoretical sophistication, and we didn't really test explicit computational models anymore for about a decade, because we had our hands full just dealing with these tens of thousands of channels. I think it was a natural progression. So perhaps a discipline that's done best in terms of explaining neuronal activity patterns with fully explicit computational models is computational neuroscience. And this is an exciting field that I'm always fascinated by and trying to follow as best I can. Computational neuroscience has been very successful at predicting sensory responses of neurons to arbitrary novel images that these models have not been trained with. So that's a major achievement, I think. And also in higher level regions, there are sort of models of evidence accumulation and decision making that sort of close the loop all the way to behavior, from perception to behavior. However, what's not completely been successful is doing this for really complex cognitive tasks. So when we look at this area here, so real world tasks like object recognition that just inherently require a lot of knowledge about the world. And I think most of what the brain does requires massive world knowledge. AI had to learn that the hard way. AI started out with ideas about very general algorithms for intelligence, and then learned that that doesn't work. They all have exponential time complexity. So what you need is massive amounts of knowledge. Initially, that was hand-coded, but then it led to the rise of machine learning. Machine learning is as important as it is, because to make intelligence work, we need to learn massive knowledge about the world. And I think in computational neuroscience also, we have to learn that we need these really complex models that don't have a very simple description, possibly. So the third highly relevant discipline here is artificial intelligence, which has been the discipline that has classically developed these computational models that actually perform feats of intelligence. And artificial intelligence is making great strides at the moment. And there is a single modeling technique that really falls at the intersection of all of these, and I think therefore is poised to provide major progress for brain science as well. And this technique is neural networks. Neural networks are artificial models inspired by the brain but highly abstracted, that can learn massive amounts of world knowledge to solve artificial intelligence tasks. Neural networks have a long history in each of these three fields. They're a very, very old idea, actually, and they have a long history in each of these fields. However, they're only beginning to be used to meet these two goals, to explain neuronal activity patterns and to explain behavioral data. I think Stanford is probably one of the best places in the world to pursue this framework, which is just emerging. And you've just made a really great decision in hiring Dan Yamins, who's one of the major proponents of this approach. So my job is going to be to try to convince you that you should be collaborating with Dan in this direction. So this is sort of the big story. I would like, at the end of my talk, for you to maybe get share some of my excitement about this direction that's currently emerging. So in order to illustrate that, I'm going to tell you a much smaller story about how in my lab we've come around to this approach and sort of have some initial results in this direction. So in my lab, we study a particular problem, which is visual object recognition. And that involves recognizing all the objects in real-world images like this one, where we do this very effortlessly. We might recognize some of the people in these images. We immediately draw a host of semantic inferences automatically. And we'd like to understand this problem. So for visual object recognition research, the goal could be stated like this. We want to build a biologically plausible network model that can recognize novel object images that the model has not been trained with, importantly, of course, otherwise it's useless, and predict their neuronal representations as well as human behavioral responses. So one really key methodological challenge in this context is how to compare representations in computational models to brain representations. This is really not an easy task because we think of the representations as inherently multivariate. We think of them as population codes. So they live in this very high-dimensional space in both the brain and in the model. And we don't know the correspondency mapping between the units of our model and the neurons in the brain that we've measured, or the voxels or the response channels that we've measured. And because of the combinatorics, this is actually not an easy problem to estimate this correspondency, or often it's estimated as a linear mapping, but there are lots of parameters to estimate, and you need a lot of additional data just to solve this kind of practical problem. We've developed a particular approach to this that I call representational similarity analysis, where for each particular stimulus, we measure a response pattern in a region of interest. So you could think of this as a little ROI and fMRI, and these would be the voxels. And then for another particular image, we have another pattern of activity here. So this is the same little region, the same response channels measured twice, but the activity pattern is different, as you see from the colors. And then we compute the dissimilarity between these activity patterns, which you could also think of as a distance in the representational space in which these patterns are points that correspond to the stimuli. We enter that dissimilarity into a matrix. So here in this matrix, you can look up for each pair of stimuli how dissimilar the two stimuli are in the representation. We interpret these patterns as representations, so we interpret these dissimilarities as representational dissimilarities. And this matrix is symmetric about a diagonal of zeros, and it gives us a characterization of the nature of the representation in this brain region. And then we can look at how this representational space changes across different areas of the brain, stages of computation. So here the color now encodes the dissimilarity. Importantly, this makes it straightforward to compare a brain representation to a representation in a computational model where we can take exactly the same approach, take the pattern across all the units for each of the stimuli, compare these patterns by some similarity measure or distance. It could be 1 minus the correlation, it could be the Euclidean distance or the Mahalanobis distance. And then it's possible to directly compare these, what we call representational geometries, by correlating these matrices. And this circumvents this correspondency problem that we have at the level of the units. So these matrices are called RDMs, or representational dissimilarity matrices. So here in 2008, we took this approach to compare representations between human inferior temporal cortex and monkey inferior temporal cortex. IT cortex is the part of the brain where high-level visual semantic representations are thought to reside. And you see that here I've color-coded the representational distances with these cold colors corresponding to small distances, these warm colors corresponding to large distances. And you see immediately that there is a strong categorical structure where all the human faces, here you see our image set of 92 isolated object images. When we compare in this representation a human face to another human face, they're always very similar, so you get this tight cluster here. When you compare an animal face to another animal face, they're also very similar. The animal faces actually are all different species, different colors. They look in different directions. So this is sort of intuitively not obviously related to visual similarity. And then there's this off-diagonal square here, which is also blue, which corresponds to all the comparisons between a human face and an animal face. So this shows that there's sort of a single face cluster. And all of this is within a larger animal cluster. And then there's also a cluster of inanimates within which the natural and artificial objects are not really clustering further. And the largest dissimilarities obtained for when you compare an animate to an inanimate object. So all of these qualitative features of this matrix were very similar in the monkey IT matrix, and actually also the within category representational geometry was very, very similar in the monkey. So we analyzed this quantitatively in this paper, but this is not what I'm going to talk about today. So after this paper kind of posed the computational challenge to us. Importantly, I should have said this human IT matrix was estimated from fMRI voxels with 2 millimeter isotropic resolution, and the monkey IT matrix was estimated from cell recordings. So despite these really fundamentally different ways of measuring these representations, taking samples of the dimensions of these representational spaces, we get in this case, a rather similar result. And that was very encouraging to us, but it's also problematic, and we shouldn't, in general, count on that. That's an interesting topic for discussion and a methodological challenge. Here, these were very, very similar. So this finding posed the challenge to us. Can we explain this representation with a computational model? So we want a model that we train in some way, and we can use knowledge about the world, we can use brain data, but we can't use these particular images, and we can't use these brain data. And then we want to predict this representational space with a computational model. And that would be a candidate for the computational mechanism leading up to this representation in the brain. So when we started this, this was the PhD project of my great graduate student, Seyed Khalek Rasavi, who's now at MIT. And this was in 2010. In 2010, inferior temporal cortex was a computational mystery, so no one had a computational model that could really predict either single cell responses to arbitrary images or representational geometries. And at the time, computer vision was most successful when using hand-engineered visual features as inputs to shallow machine learning classifiers. So neural networks had been around for a long time, but they didn't really work very well for computer vision. So we just had been aware of this problem that we don't have any compelling computational model for IT for a long time. So we just wanted something that could do the job, and we weren't picky about biological plausibility because we thought, let's have something. Let's see if we can predict these representations at all, and then in a second step we can worry about how that might be implemented with biological neurons. So we assembled these 27 computational vision models that spanned the gamut from models motivated by brain theory to computer vision models. And I'm not going to read them all for you. Some of you may recognize a few of these names. So they included simple image representations, your favorite hand-engineered computer vision features like SIFT and FOG and GIST. Most of them were untrained. Some of them were trained in an unsupervised framework, so natural images had been used to construct these features, but no category labels. And 2 of them also were trained in a supervised framework to some extent. So how similar are the model representations to IT? So what I'm going to plot here on the vertical is the RDM correlation with human IT. So that's the representational dissimilarity matrix correlation of each of the models with human IT. So it's a measure of how similar these representational spaces are. Here the gray bar is the noise ceiling. So that's an estimate of the range within which we expect the true model to fall given the noise in the data and the variability across subjects. So when we look at the result, we see that almost all of the models explain significant variance in these representational distance matrices. However, they're all very far from this noise ceiling, so they don't really fully explain the structure in our data at all. So our conclusion from this was that all 27 models fail to explain IT, and we were getting ready for writing this up as a somewhat depressing paper, but what can you do? So the models failed. However, each model explained significant variance in the representation. So a question that we wanted to address was, can weighted combinations of these features explain the IT representation? Because when you distort, when you scale the axes of the representation or linearly remix the features, of course you change the representational distance matrices a lot. So it could be that we have all the right nonlinearities present in the set. However, we have to do this sort of trivial linear transform on top of it in order to explain IT. So what we did was we fitted these features to IT using remixing and reweighting. Remixing involved fitting 3 support vector machine discriminants for the major categorical divisions that are prominent in IT. So those are animate, inanimate, face, non-face, and body, non-body to accentuate these. And then we used non-negative least squares to fit 1 weight for each of the models and 1 weight for each of these SVM discriminants to estimate a mixture that would best explain the IT representational geometry. And this was cross-validated across images so that we wouldn't overfit to our image set. So when we did this, this is the matrix that we obtained. You see that it gets this little blue square right here. This is the tight cluster of human faces, which are all frontal images and visually very similar. But it doesn't really get any of the other structure. It doesn't get the animal face cluster because presumably because the animal faces are all different species and different views and angles and colors. It doesn't get the animate cluster or the inanimate cluster. And here for comparison, you have the human IT and the monkey IT matrix again. So this approach really didn't work even with linear remixing and reweighting of the features. So then something very significant happened in 2012 in computer vision. A deep neural network trained by backpropagation improved the state-of-the-art in machine visual object recognition by a large margin. It won the ImageNet competition, where computer vision scientists submit their algorithms, and then their algorithms are tested on image sets that they didn't have access to before, so there's no way of cheating. So this is a very good way of really establishing how well different computer vision systems do at object recognition. So this was sort of a sudden win and convinced many in the computer vision community that neural networks were the way to go. And since 2012, this competition, as far as I know, has never been won by any system that wasn't a deep neural network. So for us, of course, this was super exciting, for one thing, just because of the computational advance of being able to do vision much better suddenly. And the other reason, of course, was these are the kinds of models that are inspired by brain science. They're much more biologically plausible than the models that we had been testing before as a kind of computational compromise. So I just want to give you a little bit more context on artificial neural networks. You might be wondering, this term has been around so long, what's new now about this, right? In the 80s, we were excited about this, and then somehow we forgot about them again. So what is the history of neural networks? They have a long history, roughly coextensive with the history of modern computing, I would say. In the 40s, we had McCulloch and Pitz's binary threshold units. In the 60s, Rosenblatt, Minsky, and Papert's perceptron research. In the 80s, they caused a big paradigm shift in cognitive science led by Jay McClelland, who's in the audience over there, and really transformed cognitive science in this period. However, in the 90s, people lost faith with neural networks a little bit. So in computer science, people lost faith for two reasons. The first was that they didn't really perform very well on real-world problems, including object recognition. Other methods, these hand-engineered features, beat them. And they were very hard to theoretically analyze. So people also felt that it's sort of impossible to get rigorous theoretical results, so that made them less attractive. However, more recently, there have been breakthroughs with deep learning brought about by a smaller community of researchers who kept at it in the meantime. The three most famous are probably Jeff Hinton, Joshua Bengio, and Jan LeCun, who developed pre-training and regularization techniques that enabled efficient learning of deep networks in the early 2000s. And in the last 5 years, with growing computing power and large labeled data sets via the web available, these networks have really come into their own, because now we have the amounts of world knowledge that they need to soak up in order to really work. So just in a nutshell, a unit in a network of this type computes a linear combination of its input and sends it through a static nonlinearity, which can take a number of different forms. It can be a sigmoid, it can be simply a threshold, or a very popular model is the rectified linear nonlinearity, where the pre-activation is just passed through if it's positive, and otherwise the unit outputs a 0. These simple units are then linked up in different configurations. Here you have a shallow feed-forward network with a single hidden layer. Here's a deep feed-forward network with 2 hidden layers in this case. More loosely, deep means that the network has lots of hidden layers. So it has lots of stages of nonlinear transformation, which allow it to learn more abstract representations of the input. And this seems to be a key feature of these networks that allow them to perform well and generalize well. They can also be linked up recurrently. So when the connections form cycles, these networks produce dynamics naturally and lend themselves to modeling sequences such as speech sound, convert speech sound to text, translate texts, and convert text to speech as well. So in computer vision, the kinds of networks that are dominant at the moment are deep convolutional feed-forward networks. So these have a couple of additional features that I just want to briefly introduce you to. So they take an input image, and then in the first layer, compute a convolution of the input image with a little weight template. So there's two decisions there that are inspired by the visual hierarchy. The first is that not all the possible projections to this one unit in the first layer here are present, but there's this local receptive field. So lots of units are just by definition 0, and this greatly reduces the number of weights that have to be learned. So this makes it easier to train these models. And the other decision is to say that in a given map, in the first layer, you have a retinatopic map. So this is inspired by the visual system, where the same feature is detected all over the image. And this is equivalent to computing a convolution of this little weight template with the image. And that would give you this pre-activation for the first layer for a single feature. You see that this feature looks like a Gabor. It's actually not a Gabor. It's similar to Gabor, but it's a weight template that's been learned by this network during its training to categorize objects. So this was a deep network. It was trained just to categorize objects, but in the first layer, it ends up learning these Gabor-like features, very much consistent with what we see in biological vision. So it kind of rediscovers this. So for it learns a number of different features here, just 3 shown, but it could be perhaps 100 of these or more that it learns just in the first layer. This convolution is followed by a nonlinearity. Typically, there's a stage of pooling, local pooling, for example, taking the local max or the local average. And then there's usually local normalization. And all of this together is called a single layer. And then this process is repeated with the parameters changing and the receptive fields becoming larger and larger with respect to the original image space. So networks typically will have 5 to 20 layers. Now there are many that have more than 20 layers, actually. So here's the architecture in Krizhevsky et al.'s network, which convinced so many people in computer vision that this was the way to go for computer vision. This network was supervised with 1.2 million category-labeled images, 1,000 categories. This set included lots of dogs, I think 120 dog species. I know about 2 of them, I think. And this, if you see these deep dream images from Google, people are playing around with this a lot, and it's very, very inspiring. You often see dogs, and this is the reason for it. The network has been trained with stochastic gradient descent, backpropagation. It uses dropout regularization in the final fully connected layers. And it's got 60 million parameters and 650,000 neurons in total. So a lot of parameters, despite these ways of regularizing it, having these local receptive fields and the weight sharing in the convolutional layers. So how do the layers of this network do at explaining the representational geometry in IT? So here, as before, I'm going to plot the accuracy of the human IT dissimilarity matrix prediction. So this is the same as before. I should have labeled this axis the same way. So this is the RDM correlation averaged across our subjects. So in layer 1, we get a very small correlation with the human IT matrix. However, it is a significant correlation. So there's some variance explained there, which really just means that this representation and human IT both contain visual information. It really doesn't tell you much more than this to get a significant result here. So here's the noise ceiling. So we also see that the performance of this model is really far from the noise ceiling. So this is not a good model for IT. As we go up the layers, this correlation rises further and further. And at layer 7, which is the penultimate layer of this network, we're really close to the noise ceiling. So we were really excited to see that we can get this close to it. So the deep net almost reached the noise ceiling, but not quite. So again, we asked, can weighted combinations of its units fully explain the IT representation? So we just played exactly the same game that we played with the computer vision models that we tested earlier. We remixed the features using the same 3 SVM discriminants, and we reweighted the representations and the SVM discriminant outputs using non-negative least squares. And this is the matrix that we got. So this is the first time that we saw actually sort of a model that can explain this structure that we'd struggled with for a number of years. So here again, for comparison, is the human IT and the monkey IT matrix, and we see that qualitatively these are very similar now. They have all the same categorical divisions, and also within categories a very similar dissimilarity structure. So when we looked at this model, we really reached into the noise ceiling, and I was very excited about the fact that I can now get rid of this old data set that I've been analyzing for so many years, because we're at the point now where we have a model that explains this data set. So in order to highlight the remaining shortcomings, of which I think there are many of this model, we need better data, we need more data. So this model performed significantly better than all of the other models. So we can now capture the computations of the ventral stream to some extent, at least of vision at a glance, the feed-forward component of the ventral stream. So that's very exciting, I think. However, we're capturing these computations in a very big net. There are so many parameters. We don't really know how this network achieves this, right? So we still need to find the computations in the map, in the net, but we're in a much better position now, of course, because this network is totally transparent to us. It's sort of like the dream situation for a neurophysiologist, where you can show as many stimuli as you want, and you have full data about everything in the network, all the responses, right? So this is really, I think, a very healthy challenge for analysis, because we always feel we're limited by our crappy data, right? Now here's when you're not limited by your crappy data, it's still not easy. You still have to think really hard, what are you going to do when you have the full information? And I think the very exciting circuit level developments that we're seeing actually make this a reality in terms of measuring real brains. And there we face a similar challenge. Now we have all this information, but how do we make sense of it? How do we use it to test computational theories? So I just want to show you some other people's work in the direction of understanding better what these representations are in these models. So one approach is to visualize the internal features. So here's an approach from Seiler and Fergus called deconvolutional networks. And in the first layer, we see these Gabor-like features. So I'm going to show you 4 selected units, and I'm going to show you a visualization as a template, as an image template of what this unit does. And maybe you're already thinking why that might be problematic, but hold your horses for a moment. So in layer 2, we see these slightly more complicated features of like curved segments and more complicated patterns. In layer 3, we start seeing parts of objects emerging. In layer 4, we see detectors for full objects. And in layer 5, similarly, it looks as though there might be detectors there for full objects. Now, a problem with this is that fundamentally, the reason why these networks can do vision is because they don't do template matching in the image. Template matching is fundamentally a shallow computational process. It's everyone's favorite way to imagine vision, and computer vision has shown over and over again that it doesn't work at all. So the way that this works is, for a given unit, you look for an image that drives this unit strongly, and then you look for the direction in image space along which the unit's response rises most steeply. So this is sort of a different image. So there's an image that drove this unit strongly, and this is the changes to the pixels that you would have to make to drive that unit even more strongly. So this is a particular dog, but the same unit might respond to many other dogs, usually the same category, interestingly, but it could also, from this you can't see, if it also responds to totally different categories. So take this with a grain of salt, but it gives us perhaps an idea of the complexity of these representations. Here's another exciting piece of work from Matthias Bethke's lab using a network from Andrew Zisserman's lab. They designed a network that separates content and style in images. So this is available on arXiv. It's not published yet, actually. And by separating out these two elements, they can synthesize images, which they optimize to match these internal representations of the content and the style to create these new composite images that combine the content of one image with the style of another image. And they did this for using photographs for the content and then paintings for the style. And you get these very beautiful renderings as paintings of these photographs. And some of them are really striking. And I find this extremely inspiring. And it really gives us an idea of the complexity of these models. Again, the interpretation is not straightforward, so this is sort of just something we're struggling with at the moment. In the last 2 minutes, I want to tell you quickly about predicting categorization reaction time for particular objects. I've told you in the beginning we need to also predict behavioral measures. So one approach to this is to think of categorization. When you're getting ready for a categorization task like animate, inanimate, you're setting up this internal readout filter from this rich object representation in inferior temporal cortex that gives you the optimal evidence for the object being an animate object. And we model this using the representational space in the penultimate layer of this deep neural network from Krizhevsky et al. By fitting a support vector machine discriminant. So we get a decision boundary in this deep net's representational space, and we get a decision value for each particular image. And then some images are going to be further away from the decision boundary than other images. And we predict that for these images, the evidence toward this being an animate will accumulate more rapidly, and the reaction time will be smaller. So here I'm plotting the deep net's decision value versus the human reaction time, and the decision boundary is here. So when we look at the images, we see that those images that are further away from the decision boundary are actually associated with smaller reaction times in humans. So here I've just color-coded this, and this is highly significant. So we're looking at this systematically now for different readout models and different categorical divisions, and we are interested in using this framework to have some model of the behavioral readout as well. So where are we now with this deep neural network framework? Well, there's an emerging body of work using deep nets to explain biological vision. In 2014, the first 3 papers came out that attempted this. As I've said before, Dan Yamins is an important proponent of this approach. Also, Jim DiCarlo's lab, Charles Cadieux, has contributed to this. This year, another paper from Marcel van Gerven's lab came out showing that mid-level vision is well explained by these networks as well. And there's a number of preprints in circulation on new developments, including the work from Matthias Bethke's lab that I've shown you. So are these deep neural nets like brains? Well, yes, they are. They're directly inspired by brains. All operations are biologically plausible. They have retinotopic maps of features of increasing complexity, stage-wise transformations, and they explain representational spaces, as I've shown you. But of course, at the same time, also know they have no recurrent signal flow. They have no spiking, they have no biologically justified neuron types. All this biological detail is missing, and the list is indefinite, of course, right? So is a model wrong for abstracting from a certain feature of neurobiology? I would say only if the feature is needed to explain brain computations. And so I think that's an empirical question of exactly which details we need. I think it's desirable to make bold abstractions from biological reality in this undertaking. So what's missing at the moment? One key thing that's missing is recurrent dynamics, spatiotemporal prediction, attention, probabilistic representation. Another is learning and development, other visual functions beyond mere category level recognition, and exploring, predicting a richer array of behavioral measures, categorization errors, estimation errors, sensory motor dynamics. But the point for me is that the recent advances provide us with the technological basis for taking the rich set of ideas that's in the literature and making functioning, fully explicit models that actually perform feats of intelligence. So in conclusion, deep neural nets are biologically plausible artificial systems that can perform vision and other feats of intelligence under real-world conditions. They explain low to high-level visual representations of arbitrary natural images and also predict categorization reaction times for particular individual images. So overall, deep neural nets, I hope I've convinced you, are biologically plausible models that can perform these real-world feats of intelligence, and I think they're poised to explain the brain computations that underlie perception, cognition, and action. Thank you very much.
When I stood up there. Yeah. We have time for just one question, and we'll take Daniel Fisher.
Okay, I very much wait for the microphone. So I guess
the microphone's right behind you.
Okay, so I guess I had a worry about the sort of metric that you're using for how well you're doing explaining the data in IT, and then you sort of use that and then start looking at the data coming from these deep networks. And I worry that that's rather similar to if one looked at playing chess, where at low levels, what you look at first on a board and what a computer looks at first on the board are pretty similar, and does extremely well, the computer, at predicting what move a person will do. But it plays chess completely differently by the time you get to higher levels. Mm-hmm. And so I would sort of worry whether that in some sense is going on here as well. The low levels, it is similar, but the similarity in levels above that is not exactly superficial, but is somewhat misleading.
I wonder what you mean by low level. Do you mean the categorical distinctions versus within categories?
No, the retinotopic and looking at the features that come in the lower levels of those, which clearly look biological, but at the higher levels we don't know what the biological features look like. Right? We don't know, except perhaps on these very coarse fMRI scale images.
So it looks from the evidence so far that these networks explain representational geometries across layers, also in IT, right? But you feel that because of the way that we compare the representational geometries...
Right, so the question what you mean by explaining them. You choose a particular metric and particular metrics and say one explains that. And it's not clear how connected that metric is to what the actual comparison of how they're doing things. And I was saying that may be analogous to saying, okay, you can predict the next move someone is going to do on a chessboard.
Yeah, so I mean, it's really interesting to think about how to compare representational geometries. Another approach that's widespread and that I like very much as well is to fit linear models to predict the response of each response channel to each of the images. That's an alternative method, and it's also a very good method. It requires this additional very big data set for just fitting this linear mapping, and then a separate validation data set for evaluating the responses. What you're throwing away by looking at the distance matrices is the rotation of this arrangement in the high dimensional space and any shifts, right? And so the intuition behind this approach is that these are just linear transformations that are computationally trivial, and that we want to abstract from these in order to see the forest instead of the trees. But I'd love to discuss this with you in more detail to understand more what your thoughts are.
Let me follow that thought up. We are lucky you will be here tomorrow.
I will be.
And for example, you could join us for lunch. And you can all join us for lunch. Bill, it's on Bill. Thank you very much.
For more, please visit us at stanford.edu.